for your advantage, these parameters have already been optimized. And then once you generate your read mapping, you would then go to the process of doing a local alignment. And this would proceed automatically once you just clicked the start button. And one of the first things that you would want to look at after you had done an analysis is to know how successful was your analysis. So we provide you with in-depth QC data. So what we're looking at here is the kind of coverage distribution that you have, and you can see that you have a large amount of distribution of coverage. The majority is in this 0 to 1,000, but you also have the 5,000 to 6,000, 10,000 to 11,000, all the way through the to some areas where you have coverages as deep as 65,000. And then you also can find out what percentage of your data is covered. So at least 98.94% of your coverage is present at 1x, and 95% is covered at least 100x. And then you can also see for each target that you had specified in the GeneRead panel what your level of coverage is and the specificity. So really a great deal of information to help you know that the targeting that you did in the previous part of your experiment was successful. And then another key attribute of the bioinformatics is we're going to enable you to review the history. So you can always know exactly what types of analysis that you did. So if you decide to modify any of the workflows and look back at your results, you can see what the analysis was that you performed and how you might have modified it in future. And one of the most useful and where you spend the majority of your time within the bioinformatics tools is the visualization. And in this case, what you're seeing is your reference sequence at the top. So this is the reference that you mapped your reads to. Then you can see any amino acid changes that have taken place. This region here is your mapping, and you'll notice that we are not displaying hundreds or thousands here. Even though there's 12,591 reads here, you'll see a representation of the individual reads in the top half, and then a compressed version of your reads in the bottom half of this graph, so you can see specifically where your variant is showing up, and also how well it's represented in forward and reverse reads and the greater depth of coverage. And all of these variants are linked to a variant table that you can use to navigate from one variant to the next after you've done your variant calling. So an incredibly useful tool. And we additionally provide you with bioinformatics that allow you to focus uniquely on the kinds of challenges that you might be faced with when working with an amplified sequence from single-cell analysis. And what we're looking at here is all the information specific to this single-cell analysis and this targeting. So we're looking at one of the GeneRead panels, the 101X clinically relevant tumor panel. And this is showing you the region that you've targeted, as well as all of the primers that were used to create the amplification. And when you're looking at the reads below, you can now see those reads that are in the reverse orientation, those reads that are in the forward orientation, and you can also see paired end reads. So here we're seeing one side of a paired end read, and you can see that part of the paired end read, which includes this shaded area, and this shaded area maps to these primer regions. So you can tell that the primer region here is included in the mapping, so you get the greater specificity of the read mapping, but is not included in the variant calling. So we are not artificially directed to look for variants where the primers are. And this is based on this specific functionality that is allowing us to trim primers and their dimers for mapped reads, and that's what we're seeing here. This is a primer dimer here. One of the other characteristics that one can't emphasize enough is the quality of the algorithms that one has when you are working with the genomics workbench and the biomedical workbench. These are world-class algorithms that were developed at our site in Aarhus. And we, in my saying that is typically not enough to provide you with the information that you need to have confidence in that. So we've done a number of benchmarking assays, specifically with the NA12878, or the genome in a bottle reference sequence, which has been very well characterized. So we're able to make statements not only about the true positives, but also about the true negatives, and give you some statistical information about how accurately our algorithms are able to work. So what you see here is information about sensitivity, specificity, precision, and the false positive rate. And you can see that sensitivity here, which is the number of true positives divided by the true positives and the false negatives, is very high, and our specificity is even higher. So the true positives is telling us if a variant is there, and are we able to find it, and the specificity is telling us the kind of the opposite of a false positive. This is telling us that we're finding, or we're not calling variants that aren't there. And the precision is telling us of all the variants that are called, how many are truly positive. And then you can see here the advantage of getting coverage. So here we're looking at sensitivity and the false positive rate, where the light blue bar is telling us our sensitivity in regions that don't have this minimum threshold of 200x. And the dark blue bar does have that cutoff. So you can see there's a definite advantage in making sure that where you're calling your variants is it an area that you have appropriate coverage. And this is one of the criteria that you can set within the biomedical workbench, and one of the advantages of using the GeneRead panels, because they ensure very good coverage at the regions that are important to you. So one of the things that we're able to provide to you at QIAGEN is all of these pieces together. So we, as we've mentioned, are platform agnostic, but we also have even more streamlined and more holistic application from sample to insight when we're using the GeneReader, but we can work with any sequencer. We can use the GeneRead panels, perform the analysis with the biomedical workbench, and then use an interpretation from the Ingenuity. And this gives me an opportunity to tout the knowledge base that we have access to from Ingenuity. This knowledge base is the foundation of Ingenuity pathway analysis, which has been the gold standard for doing expression analysis and pathway analysis for over a decade. Excuse me. The knowledge base provides a breadth of biological and chemical information that allows us to really see the relationships between those pieces of information because of the way that we have organized it. So you're able to see information from an individual and use that to understand their phenotype. And there you might be able to understand information about drug mechanisms and associate the drug mechanism with a cellular mechanism. And that cellular mechanism can then further be associated with a molecular mechanism. And that molecular mechanism can then be tied to a very specific RNA or DNA alteration. So the DNA alterations, or the SNVs, would be what is highlighted in Ingenuity variant analysis, and the differences in expression are highlighted in Ingenuity pathway analysis. But the real value that we're providing is, in addition to having highly accurate, highly curated data, is that we've put them together in a structure that allows you to have access to the content and relationships and take full advantage of the investment that QIAGEN's made in this ontology. And then when we put that all together in a single-cell analysis, you're able to see that the biomedical genomics workbench is going to take you through a certain point where you can take your data off of the sequencing instrument, align it to your reference sequence, generate variants, and then once you have identified all of your variants, you can now upload that to the Ingenuity variant analysis. So the point of being in the biomedical workbench is to identify true variants. And then once you've uploaded those into variant analysis, you're able to put that through a cascade of additional filters that will allow you to identify those that you have higher confidence in, those variants that you may not be interested in because they are common, such as those that are present in dbSNP or in our allele frequency database, which we are curating. You can also identify the variants that have known deleterious effects, and then use some association of the known deleterious effects with genetic information, statistical associations with diseases from the curated literature, and further associates that within a biological context. So if you know that this particular sample is coming from a colorectal cancer, the variant analysis will take advantage of that information to further filter and provide information about the relevance of the variants that you've uploaded. And then this type of analysis is possible to do at a variety of scalable workflows. So you can use this for a large number of samples, or you can use this for an individual sample. So you can use it for stratification studies, large cancer studies. You can use it for a genetic disease cohort. Typical example might be for trio analysis or other types of family analysis, tumor normal pair, or a personal genome. And for obviously, when we're talking about single-cell analysis, it would be to your definite advantage to use this to analyze common variants or shared variants amongst cells that are coming from the same biopsy, and those things that are different, to be able to know what's unique and what's shared, what are possible driver mutations, and how the populations may be changing over time. So really powerful tools that you can store and share with other colleagues. So now that I've spoken about the strengths of the tools that we're providing, I also want to spend some time talking about the two case stories that we have. And in the first example, I'm going to go over how we use the bioinformatics tools to illustrate the strength of Repligy versus Malbac, which is another method, a PCR-based method for doing the whole genome amplification. And then we're going to use the analysis tools again to look at single-cell analysis of two different colorectal cancer cell lines. So in case story 1, we're focusing on Repligy versus Malbac. And here you can see we're doing two whole genome amplifications, and that's really the only place where these two different patterns differ. On the left side, we're targeting with E. coli, just 1 picogram, and we're using the Repligy single cell kit, using that to, with the GeneReader library kit as in the other, to create a library, and then we're using the MiSeq sequencing and the 2 times 150 nucleotide version 2 amplification, and then the biomedical genomics workbench for the data analysis. So again, to emphasize, exactly the same for the two different workflows. The only difference is whether or not we use the Repligy single cell kit or the Malbac kit. And just to tell you a little bit about Malbac, this is a multiple annealing and looping based amplification kit. It was developed at Harvard. It's based on pre-amplification prior to an exponential PCR. It was developed at around 2012, and it is PCR and TAC-based, as opposed to the whole genome amplification method that we described earlier for Repligy, which is isothermal genome amplification. So everything there is taking place at 30 degrees. The first thing we'll do is we'll look in detail at the read mapping, and I'll explain to you a little bit about what you're seeing here. So again, at the top you can see your reference sequence, and we're looking at two different read mappings simultaneously. So this is in our genome browser, and you have a lot of flexibility in how you're able to configure and put data together. Here we have the E. coli reference sequence, and you'll notice we're not looking at each of the individual bases. We've turned on an option so that only the bases that differ from the reference sequence are showing up here. So you can see we have an inserted T here, we have an A here. These are paired reads, so you'll notice you have a K here, which is representing a G and a T. because these paired reads were put together and they had equal quality values for both bases at this position. So you'll see we have the T base, which matched the reference sequence, and then we also have a G, which did not, so we're flagging that with this K. You'll also notice that we have these individual lines here, which is showing you where the pairing has taken place. So you've got this read, and they are known to be paired with this link here. In cases where the pairing is completely overlapping, that's when you'd have this synthesis. So this is for the Repligy, and you'll notice in this area we have 3 different variants. And you look at the exact same area in the Malbec generated single cell amplification. And here we have many, many more variants, including some multi-nucleotide variants, showing a much greater level of diversity between the two. And since this is a known sequence, we know what the reference sequence is. That's why we chose E. coli. We're able to say that this sequence is correct, and so there should be no variants, and yet we're seeing many, many more in this Malbec-generated analysis. The other thing that's really important, and we mentioned before, is that it's important to have even coverage across the entire genome. So in some cases, an analysis might tout that it has very high average coverage, but average coverage is not good if it's very uneven. So here you can see that we have a maximum coverage of 3,000. The same thing is true here for Malbec, but we have very uneven coverage. We have some areas where it's very low and some where it's much higher. And this is at a very zoomed out level, so you can see across the entire genome. And here we've zoomed in a little bit, so you can see in the overflow region that you have much more even coverage. Here you don't even have any of the overflow coverage in this region of the genome, so it's much less than you have here. You might be curious about why we have these yellow reads here. Those are reads that have the potential to map in other locations in the genome, so those are just flagged as part of our information about the read mapping. And again, the blue reads are those that are present in pairs, and the red and the green are representing the red is the reverse, and the green is the forward. This is showing you the different zoom levels, but the take-home message here is that you have much more even coverage with the Repligy than you do with the Malbec. And so the summary story is that if you're looking in total, Repligy versus Malbec, the number of false positive mutations is much, much lower. We only had 6 insertions for Repligy, whereas with the Malbec, we had 231 different types of mutations, 222 SNP-SNPs, 6 deletions, and 3 insertions. So completely different type of error profile. And we also saw that we had a much higher of the percentage of the bases mapping for the Repligy versus the Malbec. So the next case story is focusing on 2 different cancer samples. These are cancer samples that are from 2 different colon cancer cell lines. So, and we're again taking advantage of QIAGEN's sample to insight. The only piece here that is not coming from QIAGEN is the single cell isolation that is coming from cell raft arrays, cell microsystems. But thereafter, we're starting with the whole genome amplification, which again is the really key factor here for generating quality whole genome amplification with the Repligy single cell kit. And then we're doing target enrichment with the human colorectal cancer gene read DNA targeted panel 2. This is going to amplify 34 genes that were selected based on our understanding of carcinogenesis in colorectal cancer. And then we're going to do the library preparation and sequence on a MiSeq. The data analysis is again going to be done on the Biomedical Genomics Workbench with the data interpretation on the Ingenuity variant analysis, and we're further going to take advantage of QIAGEN's expertise with pyrosequencing using the PyroMark Q24 assays for confirming some of the known pathogenic variants that would be identified in each of these cell lines. And for each of the cell lines, the LOVO and the HT29, we're doing 6 different samples. The first 2 are bulk samples, one that is done as a standard bulk sample and the other that is done as a bulk sample using the whole genome amplification to see if we're getting any bias other than the bias that we might introduce with single cell amplifications. We want to make sure that the type of results that we get from the bulk analysis is similar to the type of analysis that we get with the bulk whole genome amplification analysis for both cell lines. And then we're going to drill down and see what kind of unique and individual characteristics we can identify in each of these single cell lines that were isolated. So here you can see the first results. So what we've done is we've taken each of the key variants that we've identified and plotted them based on their allelic fractions. So we can see we have some here that are present at a very high allelic fraction and some that are present at a low allelic fraction, and these are the alleles, and we can see that for the most part the alleles that are identified for the bulk DNA are very similar to the ones that we have identified at the same allelic fraction for the whole genome amplification. So in general, we're finding that we're not introducing significant bias when we're doing the whole genome amplification, and we're ready to then take a look at using this as a method for measuring the individual cell lines. And so here we are looking at, again, each of these individual cells. Well, again, here's the bulk and the whole genome amplification for the bulk, and the 3 individual cell lines for the LOVO-3, as well as the same below for HT29 for whole genome amplification. And here we're looking at the KRAS G13D mutation, and here we're looking at a BRAF mutation. And using the DNA sequencing, we're looking at an allelic fraction of 74% with our sequencing method, and this is confirmed with 71.37%. And you'll see when we do the whole genome amplification, we have 73.3% and 67.82%. Again, for the KRAS mutation, and we'll see even tighter mapping for the BRAF mutation, where we have almost 25% and 24% here, and 28% and 28% here for the sequencing detected versus the pyrosequencing detected variants. So we're very confident that we've not introduced any bias for the whole genome amplification. So then we can take a look and see how different are the single cells. So here you can see that the percentage or the allelic fraction for this individual cell for this KRAS mutation is 67%. Here we're in the 90th percentile, around the 55 percentile. But again, you can see that the single cell amplification is consistent and confirmed by the pyrosequencing. So we have really good evidence that this is a great way to characterize the individual characteristics of single cells as opposed to a bulk amplification where you have completely different understanding of what the characteristics of the single cells are. And you can imagine that would be particularly useful when one has data coming from different tissue, primary tissue versus mutagenic sample, or metastatic sample. And once you have identified your cluster of variants, the next thing you would want to do is upload all of the variants from each of the experiments